To conduct the experiment, click on the steps one by one and follow the instructions. Take 5 grams of starch powder in a clean, dry test tube. Add 5 ml of warm distilled water. Starch powder does not dissolve in cold water to form a solution. Add a few drops of iodine solution. The solution turns blue-black. This indicates the presence of starch. To conduct the experiment, click on the steps one by one and follow the instructions. Take 1 gram of glucose in a clean, dry test tube. Add 2 ml of distilled water to dissolve the glucose and form a clear solution. Add 1 ml of Benedict solution, which turns the solution blue. Gently heat the test tube in a water bath. The blue color changes to light green, yellow, orange, and finally brick red. If the solution passes through a series of colors and finally turns brick red precipitate is formed at the bottom of the test tube. It indicates the presence of glucose, a reducing sugar. To conduct the experiment, click on the steps one by one and follow the instructions. Take 1 ml of egg white in a clean, dry test tube. Add 1 ml of 2% sodium hydroxide solution to it. Then add 2 to 3 drops of 1% copper sulfate solution. Copper sulfate is blue in color. The blue color solution now turns violet. This indicates the presence of protein. To conduct the experiment, click on the steps one by one and follow the instructions. Take 1 ml of oil in a clean, dry test tube. Add 1 ml of ethanol to the oil. Ethanol, being an organic solvent, dissolves the oil and forms a clear solution. Add a few drops of water to the clear solution. The solution starts frothing and becomes turbid, forming a milky white emulsion. This indicates the presence of a lipid, fat. To conduct the experiment, click on the steps one by one and follow the instructions. Take 1 ml of sucrose solution in a test tube. Add 3 drops of dilute hydrochloric acid. Place the test tube in a hot water bath for 5 minutes. Cool the test tube in a beaker of cold water. Add sodium hydrogen carbonate to the solution using a spatula. The solution starts fizzing. This indicates that the solution has been neutralized and is slightly alkaline. Perform the Benedict's test by adding a few drops of Benedict's solution to the neutralized solution. Place the test tube containing the neutralized solution in a water bath. The color of the solution changes from blue to green, to yellow, to orange, and finally to brick red, and a precipitate is formed at the bottom. Sucrose is a non-reducing sugar and will not answer the Benedict's test directly. The addition of hydrochloric acid hydrolyzes the sucrose to form glucose and fructose. Both are reducing sugars and give a positive test with Benedict's solution.